Hey y'all, Dr. Brown here. Let's learn about capillaries, shall we? So, capillaries are the smallest of the blood vessels in the human body. They can be usually between 7 and 9 micrometers in diameter, but they can be smaller than 6 microns in diameter. Keep in mind that red blood cells average about 7.5 micrometers in diameter, so these can get very, very, very small. Structurally, they are the simplest of the blood vessels, containing only an endothelium and a basement membrane. They don't even have a complete subendothelial layer. And these capillaries are going to come in 3 varieties, as I think you could have guessed by the title at the top of the screen that says 3 types of capillaries. So what I want to do is go through each of those 3 types of capillaries and kind of point out their major structural characteristics that give them the degree of contact between the blood space inside of the capillary and the tissue space outside of the capillary that is characteristic of each different type of capillary. We're going to begin with continuous capillaries, and they get their name because the cells, the endothelial cells, form a single continuous sheet. So continuous capillaries tend to be relatively tubular. They can contain a single and up to 3 layers of endothelia surrounded by a continuous basement membrane. Now, the cells are actually held together. They're connected to one another, or in the case of the smallest ones, you can have a single cell that's wrapped around and connected to itself, like you see in the transmission EM on the right. And the cells are going to be held closed by a structure called a macula occludens. Let's write that for you so you know how to spell it. I can get my pen to work. There we go. Very good. Macula occludens. And here's one right here. And if we zoom in and look at that structure at very high magnification, we can see that the macula occludens is actually a series of discontinuous tight junctions. This is going to form, for all intents and purposes, an impenetrable barrier. Stuff can't diffuse unless it's hydrophobic, can't diffuse from the blood space in here. So this is where the blood would be, and then the tissue space would be out here. The only way substances can get from one side of the capillary endothelial membrane to the other, or the endothelial cells, is through transcellular transport or vesicles. So what you're actually seeing inside this cell are a bunch of little endocytotic and exocytotic vesicles. So these are called micropenocytic vesicles because they're doing penocytosis, but they're very, very, very small. And this is going to give us the strictest control over contact between the blood space and the tissue space. The only way something can get from the blood space to the tissue space or vice versa is if this endothelial cell has taken it up via endocytosis, transported it across the cell, and released it via exocytosis. So this extreme vetting results in very limited contact between the blood and the tissue. And in most cases, this is what you want, especially in the brain. So we need an example of where we're going to find all of these things. And our example location for continuous capillaries is the blood-brain barrier. Brain barrier. Cool. The next kind of level of contact between the blood and the tissue space is the result of fenestrated capillaries. I'm making words. Fenestrated capillaries get their name from the Latin word fenestra, meaning window. Fenestrated capillaries have continuous layer of cells. They tend to be tubular, like continuous capillaries. They have a continuous basement membrane, like continuous capillaries. The difference is they don't have the tightly controlled contact or limited contact between the blood space and the tissue space. Instead, the cells are dotted with numerous fenestrae or fenestrations, and these fenestrations or windows are going to act like little pores. Now, they do have a membrane across them. This is not a unit membrane. You can see it right here. But these little pores are going to allow more things through than could get through via transcellular transport or vesicle-mediated transport. So fenestrated capillaries have slightly more contact between the blood space and the tissue space, much more so than we saw with the continuous capillary, but not as much as we're going to see with our last one. So our continuous capillary, our example location, was the blood-brain barrier. These are the capillaries that you find in the central nervous system. An example for fenestrated capillaries is actually twofold. So one location where we can find these is endocrine gland. And this is a situation where we want to get stuff from the tissue space made by the endocrine cells into the blood, right? Endocrine glands use the bloodstream to transport their signaling molecules or hormones. The other place where we find these is in the glomeruli of the kidneys. In this case, we want to get increased transport to get stuff out. We want to get waste products from the blood space into the tissue space of the glomerulus so that then those waste products can be excreted by your kidneys, since that is in fact their job. So continuous capillaries, limited contact between blood space and tissue space, only transcellular transport. Fenestrated capillaries, more contact between the blood space and the tissue space thanks to these fenestrations or little windows in the endothelial cell. Finally, sinusoidal capillaries are going to have maximal contact between the blood space and the tissue spaces. Unlike what we saw in the first two kinds of capillaries, the endothelial cells are not continuous in sinusoidal capillaries. They're going to have these big gaps in between the cells. Additionally, the basement membrane is incomplete, meaning that substances are free to go from the blood space into the tissue space unhindered. We're going to get maximum contact between the blood and the tissue. In fact, one other thing before I go to the other picture, if we look, we also see that these are rarely perfectly cylindrical. So if I outline the sinusoidal capillary in this light micrograph of the liver, you can see that it takes kind of a tortuous, weird little pathway. So these guys are very odd shaped and they're discontinuous, allowing maximum contact between the blood space and the tissue space. And if we look at more closely, a sinusoid, and the best example of these is in the liver. So this is a hepatocyte or a liver cell out here. Inside the liver cell, not inside the liver cell, inside the capillary itself, we can see these gaps or holes in the cell membrane through which substances can flow. So transporting across capillaries, we have in decreasing stringency of getting things into or out of the capillary. We have transcellular transport with vesicles. So vesicles are going to be the most stringent. That's going to be in continuous capillaries. Then we have fenestrae, which are much less stringent. More things are going to be allowed to cross. And then finally we have sinusoids, which are going to be the least stringent, allowing maximum contact between the blood space and the tissue space. So that's just a brief overview of the three different kinds of capillaries we find in the human body. I hope you enjoyed it and had fun. Study hard. See you later.